Okay. Yes, so we're recording.
Hi, welcome everyone. Welcome to the fourth webinar of this series. We are happy to finish with the fireworks. Thanks to our today's guest, Jessica Grahn. I think we are in the same country momentarily as I'm in Montreal airport, heading back to New York. So I'll leave the floor to Anna and to Josh, and I wish to thank Anna and Josh for the great work they have done in organizing this first series of webinars that have been mostly successful. So I leave it to you, Anna, and welcome again.
Yes, thank you very much, Vittorio. Hello, everyone again. I'd also like to thank Vittorio, who is the head of our lab in Neuroscience and Humanities lab, and to Joshua for co-organizing. And so this is the final session of our spring edition of the webinar series called Embodied Music Experience, a Biocultural Approach. And we're very pleased to present our fourth speaker, Professor Jessica Grahn. Jessica Grahn investigates how music affects our brain and behavior. Her topics include how music makes us move, how musical training changes brain structure, and whether music can benefit patients with neurological disorders such as Parkinson's disease. She has undergraduate degrees in neuroscience and piano, a PhD from Cambridge University, and is now a professor in psychology at Western University. She's a member of the Royal Society of Canada College of New Scholars and president of the Society for Music Perception and Cognition. She has received awards from the British Science Association, the Ontario government, the James S. McDonnell Foundation, and most recently the prestigious CC Fellowship. Hopefully I pronounced that correctly. So thank you very much, Professor Grahn. I'll leave the floor to you.
Wonderful. Thank you very much for having me. It is a real pleasure to be here. So yes, I'm going to be talking to you today about my research on the relationship between musical rhythm and movement, and what it is that's going on in the brain that facilitates this. So one of the things that's cool about music is that it does elicit spontaneous movement, and other sounds in our environment do not do this in the same way. So if I hear a truck backing up outside my office window, I don't suddenly find myself with the urge to tap along or bob my head. But with music, we do see this behavior spontaneously. And I just have a little video here that shows exactly how early spontaneous movement to music arises. So what that was a small playing of It's a Small World. Dun, 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 dun. And these are two 9-month-old twins. And what's interesting about this is these twins cannot walk yet, they cannot talk yet, yet they know to move to music. If you could hear, it actually seems that sometimes they're synchronized, and certainly sometimes they synchronize with each other. That is basically just chance. We know that the earliest we really see accurate spontaneous synchronization to music is around age 2. More commonly, it develops around 5 or 6. And some people just never really get to see the hang of that actually moving along accurately to music, which is an area of interest of mine. What is going on for people who don't accurately move to music, but still enjoy moving to music? So what is it that we actually move to? Well, when I, for example, have a rhythm in a piece of music, that is the first bit of information that we need to get. However, I don't actually move to the individual onsets in a rhythm. So take the famous William Tell da 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 da. If I dance to that, I don't try to capture every rhythmic aspect that's going on of da 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 da. What I do is I find this regularly reoccurring what we call pulse or tactus or salience. We spontaneously try to find this when we're confronted with rhythmic sequences, and I move to that. So now I can go da 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 da. So the beat is what enables us to move along to music. If we cannot hear that beat, we really don't find ourselves able to move. So we predict when the beat will occur, and we line up our movements to spontaneously occur with that beat. If we don't align with the beat, and if we can't find that beat, then suddenly our movements, they aren't afforded in the same way. So here's some example of stimuli that we often use. So these are much more boring than the types of music that we listen to, but they really isolate the rhythm itself. And what we try to do is keep the rhythms very similar, apart from the fact that one induces that feeling of beat a little bit more easily than other rhythms that might even use exactly the same time intervals to compose the rhythm, but do not give us that clear feeling of beat. So a typical rhythm that would induce a beat might be something like D, 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 D. And then people usually don't have any trouble, D, 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 D. We can take those same time intervals and mess them up so that the timing constraints are the same in terms of the number of time intervals you have to remember, the overall duration of the sequence, but it's just not so easy to feel that beat. Something like D, 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 D. So that irregularity means that we have a harder time processing it. And the work that I was interested in initially was, what are the systems that respond to rhythm and beat? In particular, I was not so interested in the auditory system, which is unusual for a music researcher. I was really interested in the motor system. And so what happens is if we give people these rhythms to listen to, we find a network of motor areas, including the supplementary motor area in the middle of the brain, bilateral premotor cortex, of course the auditory cortex because they're listening, bilateral putamen in the core of the brain, and bilateral cerebellum in the back. What is interesting is if we then contrast, what are the areas that respond when we have a rhythm that has a beat compared to one that does not, we actually don't see as often these sort of cortical premotor areas or auditory cortex. We actually see the basal ganglia pop out most strongly, in particular the putamen. So the putamen responds more when we have a rhythm that we can feel a beat in. This is sort of a whole brain corrected, more conservative measurement. So what we can do is we can go into these individual brain areas that we saw that responded to rhythm overall and look at what they're doing with our different rhythmic conditions. And this is just a region of interest extraction and analysis. So here the rhythms in purple, metric simple, are those that have a clear beat. Complex, you could theoretically feel a beat in them, but with the length of rhythms that we have here, people don't. So they can be considered similar to these non-metric rhythms in which the intervals are so irregular, it's not possible for any beat to be perceived. And what we can see is across all the different subdivisions of the basal ganglia here on the left, pallidum, putamen, and caudate, is that the rhythms with a beat produce greater responses than the two rhythms that do not induce any sort of regular beat. The same pattern actually holds for the SMA. So that wasn't strong enough to come out in the whole brain analysis, but when you look within that region specifically. Here we do get slightly more activity for the beat rhythms in the auditory cortex, superior temporal gyrus, but that is not consistent across studies. And interestingly, the cerebellum and premotor cortex certainly respond to rhythm, but they could not care less whether that rhythm has a beat or not. So we have this network that seems to respond more to rhythms that have a beat than rhythms that don't. And this is something that we'll be following up throughout the talk. Look at is connectivity, functional connectivity. So how much are these areas communicating with each other during different types of rhythms? So we also find that not only are the basal ganglia more active during rhythms that have a beat, we find that they are more in sync, more correlated in activity with other areas in the brain, including these premotor cortex foci, the supplementary motor area, and auditory cortex. So there seems to be something of an enhanced communication when people are feeling rhythms that have a beat compared to rhythms that do not have a beat. We've also looked at individual differences in beat perception. So we give people simple tasks where they have to try and find the beat. In this case, we actually do use music. And then we look at how performance on that test correlates with activity in the brain. And what we find here is that people with better beat perception don't show differences in auditory cortex that we can detect. Doesn't mean they're not there, but they're not as strong as the differences we do see in supplementary motor area, and then also here in the ventral and dorsal premotor cortex. So again, beat seems to be more of a motor thing than actually an auditory thing. In all of these studies, while people are having their brains imaged, they are staying perfectly still. So we ask them not to move, we ask them not to count or even imagine counting or imagine tapping. Obviously, we can't control what they do in their imaginations, but we do observe them. and make sure they're not overtly moving. And in no case have we ever seen greater activity in primary motor cortex, the part of the brain that actually lights up when you make a movement, in any of our studies. So that's not a perfect control, but it does suggest that people are not spontaneously moving, that the motor system is responding here. I don't talk a lot about musical expertise, but we do have this finding that shows that greater connectivity only for musicians between the supplementary motor area and auditory cortex. So that's interesting to us because the basal ganglia seems to be similar whether you have musical training or not, but these more cortical motor areas seem to respond or enhance their communication with auditory areas in musicians more than in non-musicians. However, I will put a huge caveat on this, which is that musicians that come to participate in our study are often very attentive. So they come in, they do the practice, we give them the practice, and they say, okay, it seems like that might have been a 4/4 rhythm. Is that right? Okay, that makes sense. And this is the button I pushed for if the rhythms are, so we have them do a discrimination task sometimes where they detect if the rhythms are same or different. So this is the button for same, this is the button for different. Okay. And then they go in the scanner and immediately we test the sounds. Like, I think it needs to be a little bit louder. I really want to make sure I can hear this well. And then they come out of the scanner and they, the trial, I think it was the 7th one from the end, I accidentally pressed the wrong button. So that one I meant to say different, but I accidentally pressed same. Compare that to how a typical person with no musical training approaches our study. They come in, they do the practice, and they go, those are kind of interesting rhythms. They sound a little like, I don't know, Morse code. And how much am I going to get paid for this again? How long is it going to take? Okay, sounds good. They come out of the scanner at the end of the study and like, okay, I was a little tired at the end, but I managed to stay awake. Do I get that picture in my brain now? And off they go. So the musicians are worried that what I'm going to do is that I've been, having all this computational power where I can actually analyze their brain activity while they're in the scanner. And because of the myth of neurorealism, that what is going on in our brains is somehow more real than behavior in the real world, they are worried that I'm going to say, well, I know your teachers have said, yeah, you're great, you're doing so well in your training, you're on your way to a healthy career, but I've been looking at your brain responses and I'm here to tell you it's all over. Your brain can't handle this. So they really invest in these studies, even though we don't say anything about their musical expertise. Even when we're recruiting, we just happen to measure it at the end, but they still care much more and they're more invested. So a lot of times, whenever you see particularly greater responses from musicians than people who have no musical training, you have to keep in mind the sort of demand characteristics and the threat to identity that comes along with that musical training label when people come in. So we don't tend to interpret these very heavily, but we do see some differences. This whole network of motor areas that are implicated in rhythm and beat perception by neuroimaging, particularly the basal ganglia and the supplementary motor area, more so for beat perception, whereas this premotor cerebellar network doesn't seem to care about whether the rhythm has a beat or not. However, all fMRI is correlational. And as I said, we can tell people don't move or tap along, don't even think about it, but we can't entirely control what they're doing. So if we really want to know if these areas are crucial for rhythm or beat perception, we need to use a more causal method. We can use people with brain damage or degenerative disease. And I have done some studies with Parkinsons and beat perception that I won't cover today, but I have published. But another method that we can use in healthy people is transcranial direct current stimulation. Oh, I see there's a typo on there that I had not noticed. So this is a causal method to alter neural excitability. You basically put an anode and a cathode on 2 spots in the brain and run a very low level current through it. Now that current is so low level, it does not elicit any neural firing. It simply alters the neuron's membrane resting potential. So this is different from TMS, which actually triggers firing. It is not terribly focal because you are running a current through a swath of tissue between the anode and the cathode, but it is selective. And by that, I mean that if a neuron is not involved in the task, altering its resting membrane potential won't change anything. So only neurons that have a role to play will have their processing, altered by changing the membrane resting potential. So if we make that potential a little bit more positive, it's going to fire more easily. If we make it a little more negative, it's going to take more input, more excitatory input before it fires. So neurons that aren't relevant to the task might have this current running through, but it doesn't do anything. It only does something if that current is changing the membrane potential of a neuron that's involved in the task. So I said we can run an anode and a cathode. Under the anode, we have excitatory effects. That is, we are making the resting membrane potential less negative, so it's more likely to fire. If we run the current the other way, this same electrode now becomes the cathode, and that slightly inhibits the resting membrane potential and makes the neurons less likely to fire given a standard input. Now, there's a lot of controversy about whether transcranial direct current stimulation even does anything. That, studies with cadavers suggest that a lot of that current gets shunted. So we have to use good controls to make sure that we can interpret what we're seeing. And one of those controls is seeing differential effects for excitatory versus inhibitory stimulation. There's no way that if the current is completely being shunted, you would expect differential effects for these. So we decided to access the SMA with stimulation. We did this because it is accessible. Ideally, we would have loved to stimulate the basal ganglia, but you can't get these core subcortical areas with TDCS. And then we also compared that to what would happen when we stimulated the left and right premotor cortex. And as I said, SMA seems more responsive to beat. Left and right premotor were active for rhythm, but didn't really care about whether there was a beat or not. And so the nice thing about this is these areas are fairly close together and can serve as a bit of a control for each other. And what we do is we have people come in and they do a rhythm discrimination task in 2 sessions, counterbalanced. In one of the sessions, they get sham stimulation. And in sham stimulation, you can see in the figure there, the current ramps up and then it's immediately ramped back down. And the reason we do that is the current ramping up is what gives the sensation of being stimulated. So people feel a tingling or an itchiness underneath the electrode and they think, oh, I'm getting stimulated. So if you had no sham session, then it could just be that we're measuring that cognitive arousal that happens when people think, oh, I'm being stimulated. But when we ramp it back down, the current no longer has an effect. And then we assign people to either have excitatory or inhibitory stimulation. And in that case, we ramp that current up again, but then we keep it on during the session. And people don't generally feel the current once it reaches a steady state because it's that change in current over time that induces those physiological sensations. A task where they had to listen to a rhythm twice and determine if a third rhythm was same or different. And if it was same, they pressed one button, and if it was different, they pressed another. And then we looked at their accuracy with the idea that if these areas are truly involved in rhythm and beat perception, then we should see better performance when people are being stimulated with the excitatory stimulation and worse performance when they're being stimulated with the inhibitory stimulation. What did we see? So for the supplementary motor area, we did indeed find that compared to shams, this is percent correct, but we also do this for D prime scores and it's the same. The excitatory stimulation in red here enhanced performance. And you can see with the individual dots there that this is a fairly consistent effect across our participants. In fact, I never see effects this consistent, so that was pretty exciting. Now, underneath the anodal, we have the cathodal stimulation where we inhibit. And what we see here is that when we inhibit the supplementary motor area, people's performance gets worse relative to sham. So indeed, this is evidence for a causal role of the supplementary motor area in rhythm perception performance. Now, when we look at the premotor cortex, we find no significant effects. So we can't conclude anything about a role for the premotor cortex one way or the other from these data. So as you can see here, excuse me, the anodal is in red in both, and it's really not very different. There are people who really seem to change, but nobody is changing consistently enough as a group. We tried to look at what some of the underlying causes of these individual differences might be. Maybe there's some sort of mixed population. We, of course, looked at musical training, and that did not predict the effects of stimulation in these studies. People with musical training tend to perform a bit better on these tasks overall, but that didn't interact with the stimulation. So this leads us to conclude that the supplementary motor area, but not the premotor cortex, significantly altered rhythm discrimination, although there were those individual differences for the premotor cortex that we would still really like to investigate. So this provides converging evidence for a crucial role of the SMA and perhaps interconnected areas such as the basal ganglia. So perhaps the information that's being received from the basal ganglia is processed more efficiently by the SMA, or input from the SMA to the basal ganglia is more effective. So we can't say that it's solely the SMA, but the SMA-based network is involved in rhythm perception. So one of the areas of research that particularly the students in my lab have been driving forward is whether we can use this, information about how the motor system is responding to rhythms for good. And so, because scientists are very insecure people and we don't like to think that anybody can just understand everything we're doing, we call this particular type of therapy I'm going to talk about rhythmic auditory stimulation, and we use this for disorders in which motor areas are commonly effective. All that really means is playing music or a metronome to people while they're trying to walk and measuring what the effect of that is on their movement. This has been most commonly used for Parkinson's disease, although we have done some studies with stroke, as have others, and Huntington's is another area I'd like to look at, because Huntington's also is a basal ganglia disorder, but it predominantly affects the caudate initially instead of the putamen initially. So there are other areas in which people try this. In Parkinson's, we have difficulty particularly in initiating movement, and also we may have freezing where people are walking and then suddenly they stop. Their body stops responding to the command to move, and this is often triggered, for example, by a doorway or passing somebody. And as you can imagine, if your body is moving forward and suddenly stops, one of the things that can happen with freezing is falls. And so we really want to work on that. They also have a more unsteady gait, sometimes shuffling their feet to keep their balance. And we know that their basal ganglia activity is altered from the death of dopaminergic neurons that feed into the basal ganglia. So there's no sound in this video, but this just shows the effects of one study that was published on gait. So this is a Parkinson's patient walking with nothing in the background, no music or metronome playing. And their job is to walk through this virtual doorway that you can see cut out here, and to touch a button on the other wall and come back the other way. So I'll just play that. And you can see the patient is frozen here. So they managed to turn, they're trying to initiate that gait to carry on walking, but they aren't able to. And now this is the same patient, but this time instead of walking in silence, they're walking with a metronome. So just a tick, tick, tick. And that metronome has been set to 10% faster than their baseline walking cadence. And you won't hear the metronome here because it's not in the video, but you can see the change in the gait that results. Oh, it's the same video. I'm sorry. Here we go. This is with the metronome. I was wondering why that was happening. So as you can see, the gait is dramatically improved when this person was listening to a metronome. So why don't we all just walk around listening to metronomes all the time? If we have Parkinson's, this clearly is very useful. And the problem is we have, although it's promising, it's very variable across individuals. This is partly as a result of, as often plagues any research with neuropsychological populations, small n's. We also have variations in the stimuli that are given. So sometimes we use music, sometimes we use a metronome. Sometimes the experimenter fancies themselves a bit of a composer, so they create the music. Other times, participants bring in their own music. Sometimes that music is manipulated to enhance the beat. So there are all kinds of differences that occur. And different studies will find effects on different parameters. So some are very interested in stride length, some in stride velocity, some more in balance or in the timed up and go task, the time it takes a participant to go from seated to standing and walking. So if you look across studies, there is evidence for a reliable but small effect of auditory stimulation on gait. But part of this variation comes from all the different parameters that people look at. So one way to improve this would be to try and understand the underlying mechanisms. So we actually still have no idea how this works. There are a lot of theories. I've put some out there too, but we really don't know what that is. So there are multiple potential mechanisms, and they're not mutually exclusive mechanisms. So it could be that we hear a rhythm and we have a sort of remediation of an underactive basal ganglia, that it stimulates the basal ganglia directly, and so it affects that deficient circuit. Or it could be that the cue activates a compensatory network. In particular, people have proposed the premotor cerebellar network because we know that this network is very involved in responses to external cues. So maybe we're bypassing this medial basal ganglia problem by using another network. Or maybe it's nothing to do with that. Maybe it's because we like to listen and move to music. And so it's the reward system. And dopamine in reward-related structures is preserved for longer in Parkinson's than in some of these motor structures. And so maybe it's just that people enjoy this, and that is facilitating their movement. Or maybe for different people, it's different aspects of these things. Or maybe it's all three. So we really have no idea. So here is a very rough schematic of some of the connections we might look at, and how, for example, if we were able to do neuroimaging, we might predict that the remediation pathway would come through where auditory cortex input comes through the thalamus, increases striatal activity, which leads to reduced inhibition through the globus pallidus, which results in greater excitation through the rest of the cortical motor output network. On the other hand, if we are actually going via the cerebellum, and that's really what's key, the cerebellar premotor cortex, we might see changes that way. Or if it's reward, then we might see nucleus accumbens activity really driving this. So there are different possibilities here about what might be going on. In addition, the mechanisms of RAS may differ based on what the different musical features are that we're using. So maybe that's why we see such variable effects. So for example, maybe different musical features, for example, groove or enjoyment, groove is just how much the music makes you want to move, might really activate the reward network, that we like this music or we like moving to it. Whereas beat salience, having a very clear beat, might be something that is more from that compensatory external cueing network. And this individual variability may reflect different contributions of these different mechanisms. For example, if you are a very good beat perceiver, maybe your compensatory external cueing network is doing a lot for you. If you're a terrible beat perceiver, if you have a hard time feeling and synchronizing to that beat, maybe what you really need is something more reward-based, so something that you really enjoy. So there are lots of different possibilities here, but this is why doing this kind of research is complicated, because the music itself really varies, the underlying mechanisms vary, and then people vary. So with all of this variation, we've been trying to drill down and start to ask whether these different things affect movement differently. So we have a gait lab. We actually have a bigger gait lab here than is shown here. And one of the things we also use is a pressure sensitive walkway, instead of having our participants have to wear a lot of kinematic monitoring. This allows us to measure what we call the spatiotemporal parameters of gait, the aspects of gait that unfold over space and time, which are things like center of mass, stride velocity, stride length, cadence, so how many steps per minute you're taking, and variability of these measures as well. And so what we usually do is we have people walk on this walkway, and they walk back and forth several times. And we might, for example, instruct them to synchronize their walking to the music that we're going to play, or we might say, oh, don't worry about that. And what we might predict is that, for example, people with good beat perception will have no problem whether they're instructed to synchronize or not. But if you have poor beat perception, maybe instructing you to synchronize is actually making you worse. So what we want to see is whether these things interact. And now I'll see, I've moved my speaker a little bit. So I'll just see, this is our little test of beat perception ability. If it works, you'll hear music, and then you'll hear beeps on or off the beat of the music. And your job is to say whether they are on or off. So let's see if this goes.
Yeah, we kind of heard it at the beginning, but now it's gone.
It went away. Okay. All right. So basically you have music and you have little beep, 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 and you have to say if it's on or off. And people with good beat perception find this task quite simple. People with poor beat perception really struggle. And there are some that are harder examples than others. So we have a whole range. And so we give people this test, and then we examine whether that affects their responses to walking to music. One of the things that we have found consistently over and over again is that musical groove matters. And as I said, groove is just how much does this music make you want to move? So this is a graph of stride velocity. We give people music that is low in groove and high in groove. Now I'll talk a little bit about the music selection. So what we do is we have little short clips of music that people rate. And they rate them on how much it makes them want to move, but they also rate them on things like enjoyment or familiarity, or how clear they find the beat. And then what we try to do is equate our stimuli on these other three factors so that we're manipulating groove by itself, so that we don't have this problem where enjoyment is also different between the low groove and the high groove stimuli. Now here in this graph, we see that low groove music does not change your stride velocity. So 0 is the rate at which they walk at baseline with no music. And then if they go higher, that is a proportion improvement or speeding up in stride velocity. So we see this pattern in younger adults, older adults, and also people with Parkinsons, that the high groove music substantially alters gait more than the low groove music, and this is very reliable. Very surprisingly to us and to others, enjoyment seems to have little effect. So we just ask people how much they enjoyed the music. And as I said, we try to control for groove. So there are pieces of music, people often ask how we do this, where you can't help but tap your foot, but you really don't like this piece very much. And so often that's where the low enjoyment but high groove, or higher in groove pieces come from that we can choose. And then it's very easy to find music that you enjoy a lot, but that is low in groove. Some people talk about chill out music or ambient music, calm or peaceful music. These tend to be the sorts of things that are lower in groove. And some of that you may enjoy and some you may not. And what we found is that enjoyment really does not make a difference. So here we used music that was overall higher in groove. So we didn't manipulate high versus low groove, and we chose mostly high groove because we wanted to see if enjoyment would have an effect on top of that groove. And so we see that people are walking faster, and this is true for younger and older and people with Parkinson's, but their rated enjoyment of the music does not seem to cause any reliable difference. Now, it's possible if we had a stronger manipulation of enjoyment, you bring in that music that you love, that, if it plays at any wedding, you will definitely get out on the dance floor. The problem with asking people to do that is, although it does produce bigger enjoyment effects in terms of how much people rate it liking the music, it also has all of these other confounds of familiarity and personal relevance and this sort of stuff. And our enjoyment ratings did differ we had a 100-point scale, and the high enjoyment was about 60 points higher than the low enjoyment on that 100-point scale. So we were successful, I think, in a face valid way of manipulating enjoyment, but this really does not seem to have an impact on how quickly people walk. So what we've also looked at is beat perception ability and how that interacts with telling people to just walk freely or to synchronize. And what we find is that this is just for younger, healthy adults, that interestingly, we didn't get the interaction we predicted. What we thought might happen was that, If you walk freely, people with poor beat perception would suddenly be released from this dual task of having to synchronize, and they would start to walk faster. But that when they had to synchronize, this would be difficult and require a lot of focus, and they would slow down. But what we actually see is that synchronizing speeds everybody up, whether they have poor beat perception or good beat perception, compared to free walking. However, poor beat perceivers absolutely show less of an effect overall than the good beat perceivers, and that is significant in the synchronized, but it turns out, can't remember if it's reliable in the free walking. It's clearly smaller. And you can see when we don't have people synchronize in this study, the younger adults, they aren't really that much different from baseline. So this does certainly explain some of the individual differences that we see, that some people who have poorer beat perception are not responding as much to the music that we play. Now we can look at Parkinson's as well. Oh yeah, so this, let's see. So this pattern is also similar for older adults, except that overall older adults show much less benefit. So it's still the same that poor and beat differ in the same pattern they do here, but the overall speeding up is less for older adults. And when we look at Parkinson's, when we ask them to synchronize, first of all, we can note that actually everybody is slowing down in our Parkinson's group, regardless of whether they are good or poor beat perceiving when we ask them to synchronize. The poor beat perceivers are slowing down more than the good beat perceivers. So clearly doing this task is not beneficial when we're asking them to synchronize, and it's more harmful if you have poor beat perception. However, this very counterintuitive effect that we had, which I would like to replicate before I put all my eggs in that basket, although this was a group of about 22 Parkinson's patients, so it wasn't a tiny group, is that when we said walk freely, our good beat perceivers still stayed at about baseline, but now our poor beat perceivers sped up. So as I said, this is something to replicate. It's kind of along the lines of what we predicted in the sense that poor beat perceivers would benefit from not having to synchronize, but we really didn't expect good beat perceivers to not benefit, either with synchronization, which we show here, or with free walking. So that the fact that good beat perceivers were really just not responding much at all to this intervention is not what we predicted. So that will be something for us to follow up for sure. The take-home messages are that groove absolutely alters stride velocity. If you listen to music that makes you want to move, you move faster. The enjoyment effects are very minimal. We really don't see an effect of enjoyment in our work, and that instructions to synchronize can help or hurt, and that this depends on your beat perception ability and also what population you are looking at. Now, so the future work, as I mentioned, I'm very interested in mechanisms, and we've been interested in trying more virtual reality, as a lot of people are, in particular combining fMRI with virtual reality walking. So you can use foot pedals to navigate a sort of virtual environment in the scanner, and people with Parkinson's do freeze in those virtual reality environments. So although, of course, lying down in a scanner does not mimic all of the aspects of gait we might be interested in, certainly some of the effects in terms of the body responding to commands to move are replicated with Parkinson's patients in the scanner. And we're also just interested in looking at this too, in terms of if we're playing different musical cues, what do we see in all populations in terms of those different networks that I talked about? So yeah, we would like to look at auditory motor system changes during walking across those different musical features and populations. The other thing we've been doing, this is a picture of the new gait lab we have, which is a bit bigger, is we've been trying to use portable functional near-infrared spectroscopy with walking. So they're actually walking and looking at what is going on when people are walking without music and when they are walking with music. We are still in the very baby stages of looking at this, just trying to see if we can get good data while people are walking. We're using the portable FNIRs you can wear in a backpack, and then you have this cap on your head, as shown here. And so some of the stuff that we would be interested in doing here, if we can get reliable data, is looking at the short-term responses to music, but then also what happens if you do a longer-term intervention, so giving people multiple sessions of this. And in particular, we would be interested in combining some of these longer-term sessions with transcranial direct current stimulation to see if we can enhance the interactions that happen between auditory and movement systems by using TDCS to sort of increase the potential for these changes to stick. There's a variety of evidence from other domains that show that TDCS is often most effective when it is used in training studies, especially those that have several sessions. And then finally, the last bit of research that I'm interested in that we've been investigating is looking at non-human animals. So we can't do a lot of invasive work with humans and their auditory motor interactions, but we can with animals. So this is a video I don't think you're gonna be able to see of what a monkey does when it's asked to pull a lever in response to a beat. Yeah, I can see it's not picking it up. So I can tell you what happens is that the monkey hears the beep and then pulls the lever. So it's sort of beep, pull, beep, pull, beep, pull, beep, pull. Whereas humans, if you say, do something with the beat, they will time that movement so it happens in sync, even if you don't instruct them to. So we're really interested in what's going on in the differences between human and non-human primates. And what we have done as a first pass, this is some fMRI data from a macaque monkey, and just listening to rhythms. We haven't looked at the beat versus non-beat yet, just to see, do they show the same motor system responses that we do to begin with, in which case that suggests that the motor system really is a fundamental part of our timing systems for sequences. And indeed, on the left here, you can see very clearly outlined the putamen of the basal ganglia of the macaque. And then on the right, you can see auditory cortex. This was partly just a data check to make sure that we were getting something. So that is interesting. What we'd like to do going forward is actually look at this beat versus non-beat comparison and see if, even though they don't move to the beat the same way that we do, whether they still show these systems that are sensitive, because there are lots of reasons why animals might not show the same behavior as us. And this is one of the actual good uses of neuroscience that isn't just look at my cool data, is when an animal or a person like a baby can't tell you behaviorally, or you can't find a good behavioral index of a certain capacity, you can compare brain responses across species to examine whether they are showing sensitivity, for example, to rhythms that have a beat versus not. So in some take-home messages, rhythm perception not only activates, but also relies on our motor areas. If you alter motor activity, you alter people's rhythm perception abilities. And the effects of music on movement really depend on musical features, on beat perception ability, as well as the instructions that you are giving people. So this is a now already slightly out of date picture of all the people who are actually doing the work in the lab, while I get to sit here and talk with you guys about all of it. So I would like to acknowledge all of their hard work, particularly during COVID, where I do think students really had it pretty hard trying to pull off a research degree with COVID. And even now, we're not out of it yet, is really difficult. And so you all deserve a big pat on the back. And thank you guys all very much for your attention.
Thank you very much, Professor Grahn, for the stimulating, pun intended, talk. And I'm sure there's some questions in our attendees that they would like to ask. And so let's open up the floor to discussion. I see Vittorio has one. So Vittorio, please.
Yeah, thank you so much for your fascinating talk. My question deals with what do we know about the cross-modal or multimodal nature of this mechanism? So to put it more directly, what happens if we study rhythm or beat using visual stimuli? So squeezing a blob of lights or stuff like that. Can we entrain the same circuit? And second related question, for poor beat perceiver, can they enhance their performance if we combine visual and auditory stimulation?
That's very good question. So we have done some work with visual beat perception, and we find it is much harder to induce visual beat perception than auditory, but you can do it if you really optimize your stimuli. So flashing lights are not so good, but for example, things that rotate, or in the days of graphic equalizers, where you had a sort of stimulus that would go up and down in response to the beat, people are then much better at synchronizing. However, the visual system, even just the temporal resolution of the visual system, is worse than the auditory system. So what we find is that if we try to use visual stimulation to help poor beat perceivers, it doesn't seem to help very much, because it doesn't seem to be that their auditory system has a problem, but the visual is much better, that they are still relying on the auditory system. So we haven't found much enhancement with the visual. However, some very recent research I just submitted that we've been looking at, we've been using a video where somebody does, like a stick figure does deep knee bends. So it's sort of bouncing up and down. And then we find that people's entrainment ability to a visual stimulus is more effective. So when you're mirroring movements that somebody else is making, that's very different. We don't tend to use those sorts of stimuli. The closest I can think of is Jasmine Sue, I believe, in Munich. She has done these great point light displays of people dancing, and you can really see the rhythm in their dance movements then. So I think if we go to these more embodied types of visual display, that will be a real game changer. That's a different kettle of fish than a flashing light.
Thank you.
Just a reminder, if you have a question, please raise your virtual hand and I'll activate your microphone. Joshua.
Sure. Thank you, Jessica. It's a fantastic talk, and it's all really interesting. I had a specific question about the study in which people were walking to a musical stimulus, and they tended to speed up when they were instructed to synchronize or encouraged to synchronize. And I was just wondering, with the stimuli, I sort of assume that the tempo of those stimuli must be faster than their kind of natural sort of resonant frequency walking rate. And I was wondering if you tried different stimuli, perhaps slower than what they might have wanted to walk at anyway.
Yes, that is a key thing that I forgot to mention, is that what we do is we take a baseline measure of people's walking, and then in the data I was showing you, we sped it up by about 15% in terms of beats per minute. Usually the velocity doesn't quite reach 15%, but sometimes it does. We have a set of studies where we leave the cueing at baseline rate. And there you have a bit of an issue that, like, sometimes people do walk faster for sure. But then you have this problem of determining whether they're synchronizing or not becomes difficult, because you have set the stimulus to be at the rate that they would normally walk. And so now the only measure of whether they're synchronizing or not is a phase measure. And phase data with gait are tricky, because although people often use heel strike to say, okay, that's when the person intended to synchronize, we actually don't know if that's how people walk. They may be using, for example, the shift and balance or the toe off. So then it's like, oh, well, were they out of sync? Were they out of phase? Or were they just synchronizing because gait is continuous to a different aspect of the gait cycle? So the speeding it up at least helps us detect a little bit when we look at, steps per minute, for example, whether people are successfully synchronizing, because that is another data check that I didn't talk about, but that we need to do. Of when we ask people to synchronize, some people do well and some people don't. And so does that have an effect? And I didn't talk about that today, but that's something we look at. 10% faster is sort of what we're settling on. Other studies have looked at slower and faster, specifically with Parkinson's. In fact, there was a large study where they had Parkinson's patients come in for 3 times a week for a half-hour walking session. And at the beginning, they decided whether to play it faster or slower based on whether in the first session, gait seemed like it improved more with faster or slower. And it ended up being, I think, about half the patients, it was better doing faster, and the other half, it was actually better doing slower. So there are definitely things that, like in younger adults and healthy older adults, we don't tend to see, if we slow it down, they tend to walk slower. If we speed it up, they tend to walk faster. But that might not be the case with people with Parkinson's.
Another one from us, Annie.
Hi, Jessica. Fantastic talk, and thank you so much for sharing your findings. Just curious, very simply, about your virtual reality stimuli. So what stimuli in VR are you using? And then could you explain the role of audio or music in your potential study? Thanks so much.
Absolutely. So right now we're doing something outside the scanner just to look at VR. The main benefit of VR, I think, clinically will be that we can mimic an actual environment that isn't a lab with obstacles and things you have to navigate. So we can create a far more realistic situation for people's walking. Right now, though, we're actually using it to do some of this audio-visual comparison. So we have made, it's a very boring VR stimulus, but it works. And what we're using the VR for is to have an avatar. So that's the real benefit. So the stimulus itself is like a flat field with like a cobblestone pathway or whatever, and a stoplight at the end. And what people do is they walk in loops on this mat and then make a circle. And so, you know, that you don't need VR for. But what we've been doing is having an avatar walk in front of them, and they try to synchronize with the avatar. And the avatar is either just visual, so their footstep sound has been, you know, disappeared, or you have the avatar and the avatar's footsteps, or there's no avatar and you just hear the footstep sound, you know, of somebody and go. This has been very entertaining. So for one, just dealing with the VR parameters of you make a VR avatar walk faster. They do this funny thing where like they kind of glide through their footsteps a little bit. You know, they're not perfect biological motion parameters. And so you can speed up the gait parameter of cadence, but it will have effects on stride length as well. And so there are these funny things that we've had to deal with when we're trying to match the avatar's cadence to the person's. But the funniest thing is what we do is we'll play the avatar, make the avatar walk like 5% faster or 5% slower and see what happens. And we had to, so initially we had people do 8 loops around, but what would happen is if, for example, they weren't keeping up with the avatar very well, the avatar would lap them and then potentially walk through them. And this was so aversive to some people, it's very disturbing. So you have these social effects of somebody like walking next to you, or like you can sense that they're gaining on you and you speed up. So, and that wasn't really our intention, was to like test solely, you know, or not, you know, we wanted to look at what was happening in terms of mimicking someone else's movement and whether that would be effective, but we didn't want this like social thing of like, oh my God, they're getting closer and I'm gonna have this weird experience, or like people will slow down much more so that they don't get too close to the avatar as well, which is fine if it's a synchronized condition and you want them to, you know, match the movements, but if you don't want them to synchronize. So we found that if we just shortened the number of laps that people did on any given trial, usually the avatar didn't catch up with them and they didn't catch up with the avatar and it was okay. So that's been what we've been trying to do is, you know, can we model movement for people using virtual reality to enhance some of these auditory effects? So yes, that's been a whole ball game of entertainment, and you know, at the moment things are not as controlled as ultimately you might like. On the other hand, sometimes with therapeutic interventions, it doesn't necessarily matter. If you want something that works and it works, great. So anyway. In the scanner, what's really useful is that we can get that sense of navigation. So there, we're just creating, you know, sort of like these first-person perspective games that people have. Because if you just have people walking on foot pedals, it's just an arbitrary foot movement. But if they have to stop or turn or you know, go through a doorway, then in the scanner you can, you know, make that walking feel a little bit more realistic. But we haven't thought about doing the avatar in the scanner yet. There might be too many technical hurdles there. Maybe we'll get there. Oh, Anna, you're muted.
Sorry, Denise has a question and you should be able to use your microphone and camera if you like now. At least Denise raised her their virtual hand, but yeah, maybe you already answered that question. Okay. Oh, sorry.
Oh, I think Denise, do you want to try typing your question if the microphone's not working and someone can read it out?
Should be working. Also, I've made you a presenter now, so now you should definitely be able to speak. Is there something in the chat? Doesn't work.
Hi, thank you for the presentation. One of your findings got my interest, especially when you said related with groove, there is no effect of, or like little effect of enjoyment, whereas previous groove literature, enjoyment highly correlate with wanting to move. Could you elaborate more on that? I would like that. Thank you.
Yes, absolutely. So this is why we have to try and control for these things, because they are naturally so correlated. So it is a little artificial when you try to manipulate enjoyment and keep groove the same. And in fact, in that enjoyment study, it still wasn't perfect. There were slight differences in groove with the differences in enjoyment in terms of the ratings. However, even with those slight differences in groove, the enjoyment stayed very flat. So whatever it was, you know, we got away with it. But yes, I think this in the real world, these things are, you know, groove is a pleasant thing, right? Wanting to move to music is nice. And so most of the time, those things would go together. For us, it was more about trying to separate out the theoretical construct, in part because clinically, if the enjoyment part really adds more to the groove, then we really want to make sure that we don't use music that I bring in, for example, that they really choose it. The other thing, if you turned it around the other way, is that the good news about enjoyment seeming not to have much of an effect one way or the other is that it means people can use stuff they enjoy. And so they're more likely to use it if it turns out to be therapeutically beneficial, if they like it. So it could have been that high enjoyment slowed people down because they were too busy, you know, rocking out to the music in their heads instead of walking. We didn't expect that, but it's theoretically possible. So I would say what this suggests is that like groove really is the primary thing. If it also comes with changes in enjoyment, you don't have to worry about that. But yes, it is a job. We have to present lots of stimuli. And often what happens is like you have country music. It's like a genre thing, or metal music where the beat is super clear and people do find themselves like, yeah, I kind of want to tap to this, but they don't really like this genre very much. They don't enjoy listening to it. And so that's how you can get these differences. But yeah, it is absolutely, you're completely right that those are normally very tightly coupled.
Well, looking forward to reading more about that. That's an interesting point. Thank you.
Okay, Felix, I'm activating your...
Thanks. The mic is
and your camera. Yeah, as well, if you like.
Okay. Yeah, thanks, Jessica. I'm learning a lot today, so thanks. I think there are two questions I had is, when you use stride velocity, why do you use this measure? Because, for example, in Parkinson, people used to measure stride stability, for example, and why velocity? Like, why walking faster would be a proxy of better walking?
Yeah. No, we have all of the other metrics as well. So it is purely for the simplicity of the talk that I just present one metric. But yes, so we have stride width as well as double support time, variability of these measures, cadence versus length. And indeed, you don't want to speed people up, but actually make them more likely to fall, right? That is not a good intervention. So yes, we do in our papers report all of these, and our reviewers hate it because they're like, you're testing like 12 different metrics. I was like, well, they're relevant, you know.
Did you see the same effect of groove, for example, on stability, on something more, variability of the walking?
Yeah, so what we find is that stability itself doesn't seem to change that much, which is kind of good. So you wouldn't want them to walk faster and be less stable. So for example, stride width does not increase, which we're happy to see because it means that people feel okay while they're doing this. And then the variability measures, we don't see very much, to be honest. We do between the synchronized and the free, but we don't within the different groove conditions.
Different groove. Okay, and the last thing I was not sure to understand is whether you fixed the tempo. You talked about tempo with Joshua, but was there variability of tempo across high groove and low groove or not? Because could also be, like, would be fun to see if you make them speed up just because they enter into something faster, or also because you tap into something more groovy and more rewarding, and then maybe you could disentangle the two factors like tempo and groove.
Yes, disentangling tempo and groove is a good idea. Right now, we equate the tempo of both. So we have people have other data with tapping that shows that this is where the beat of the low groove is. Yeah, so right now it's all tempo equated, but I think tempo is quite a salient factor for sure. It's important.
Okay, all right, it's clear. Thanks.
Okay, Yeah, I'll ask a question if there's a free spot now. Thanks for your talk again. I was wondering if there's any literature on musicians or dancers developing neurodegenerative diseases that affect the motor system, like Parkinson's, and how such individuals fare compared to non-musicians or non-dancers.
This is a great question. I have wanted to do like a hospital-based study where we just look at, for example, prevalence of music and dance training in Parkinson's versus some control other diseases. And I've never figured out, you know, who I need to collaborate and how to put this together and whatever else. But and of course there are all these confounds, so that's why you had to pick your control things carefully. Of like, musicians and dancers usually come, at least in, you know, North American culture, from higher socioeconomic status because they have the luxury of access to this sort of training and all that kind of stuff. But I think this is really interesting. I have a couple students who are super interested in dance, so we're actually going to do some dance interventions where they're taking a class already, the sort of dance in Parkinson's, and looking at before and after. And what we'd really like to do is, while they're doing some of these, you know, maybe do some TDCS to see if that enhances the effectiveness. I can say that the people who have prior music or dance experience tend to be the ones who seek these kinds of opportunities out. It's not solely that, for sure. So the fact that you feel comfortable accessing that kind of therapy is good, but I don't think we know anything about progression or prevalence based on your training. But I'm with you. I think this would be such an interesting thing to look at.
Thank you. Okay, well, if there are no other questions, if you think of something, please write to Professor Grahn. Otherwise, I think we can just thank Professor Grahn for, again, a really stimulating talk and a really nice finish to the series. Thank you very much. Thank you again to Vittorio, to Joshua, to all the attendees and all the speakers. And have a nice spring, and hopefully we will have another session soon. Seeing as there's definitely more to talk about.
Wonderful.
Stay tuned. Stay tuned. Thank you so much. Bye-bye.
Bye.
Bye. Bye, everyone.
Thank you. Bye.